Good morning, students. This is your teacher Neela Mehlawat, and we'll today start with the next topic, that is introduction to the transcription. Okay, this is a very important topic. Like as we had replication, this is also one of the important topics, and it is being in board. Many questions are being asked from this topic. In general, this chapter itself is very important. Okay, clear? So should we start, everyone? Keep your books, pens, and everything ready. Shall we start, everyone? Okay, let's start. Now, first of all, let's talk about what is transcription. Okay, what is transcription? We've talked about the central dogma, isn't it? We've talked about the central dogma. We know that there are three processes that will be occurring. One is replication, isn't it? Second is your transcription that I'm talking about. And third is your translation. This is another process. Okay, so replication, transcription, and translation. Now replication we've talked about. Now transcription means we will synthesize RNA from DNA. We will synthesize RNA from DNA. Okay, let me erase so that you can clearly see. In this process, RNA will be made from DNA. So this process of synthesizing or producing RNA from DNA is called transcription. So what is transcription, students? The process by which RNA is produced from DNA. I hope the term is clear. Okay. Now talking about the RNA. Let's talk about RNA first. What is RNA? Some features of RNA. RNA basically is also polymer of ribonucleotides. See, if you remember, DNA is also polymer of deoxyribonucleotides. You remember? So DNA is a polymer of deoxyribonucleotides, while RNA is a polymer of ribonucleotides. Polymer means it has a repeating unit. It is mainly made up of ribonucleotides. Okay? RNA is made up of units of ribonucleotides, and these units, they are held together by phosphodiester bond, and it is single stranded, and the direction of bonding is from 3' to 5'. So RNA is basically made up of ribonucleotides, and they are held together by phosphodiester linkage bond, and the direction is from 3' to 5', right? Secondly, the DNA molecule, it helps in storage of information. It helps in transmission of genetic information. ये information को store करता है, ये information को transmit करता है. It passes from one you can say generation to another. All RNA except the RNAs which are present in certain viruses, right? All RNAs except a few which are present in certain viruses, they're all derived from the DNA. Means all the RNAs, they're always produced from DNA except the RNAs present in certain viruses. Because in certain viruses, RNA is a genetic material. They do not have DNA. Okay. Now we have three kinds of RNA. There are three kinds of RNA that is being produced from the DNA. One is mRNA. mRNA means the messenger RNA. What is mRNA? mRNA means the messenger RNA. Messenger. Okay. It transfers the information from gene to ribosome. So information को pass करता है gene से ribosome में. Second is tRNA, that is called transfer RNA. So it reads the mRNA, and transfers the amino acids to mRNA. ठीक है, ये उसको read करता है, and whatever information this mRNA is required, tRNA will bring it. Then is rRNA, that is ribosomal RNA. So it is constituent of ribosomes. It helps in making the ribosomes. Okay. Other than this, there are many additional RNAs which are helping or which are performing as an enzyme. So they have a catalytic activity. They have certain regulatory functions in the cell. So in totality, we can say there are only 3 types of RNA, majority. Okay, 3 types of mRNA, mRNA, tRNA and rRNA. Okay. Now see, this is the process I have depicted in case of diagram. DNA will make all kinds of RNA. It will make RNA, it will make mRNA and tRNA. And majority of the RNAs, they are constituted by these 3, tRNA, mRNA and rRNA. Okay, other than this, there are 3 other RNAs are also found. One is miRNA, ribozyme and U1 RNA. But mainly we won't be focusing on these 3. We won't only focus on these 3. So we'll concentrate only these 3. We should only remember these 3, right? Now in replication, as we've talked about, replication means producing DNA from DNA. So in replication, complete DNA molecule is copied. Okay? But in case of transcription, only portion of DNA, like particular gene or a set of or group of genes are copied at one time. And some portions, they are never copied. So in case of transcription, there is some selection that we will copy this gene and we will not copy this gene. But in case of replication, the entire DNA is being copied. Okay. So here only we will only copy a few segments or portions of the gene, right? Now we must remember what is a gene. See, we have spoken this earlier also, but I am revising it for you. The gene is a segment of DNA. Gene is a segment of DNA and it is made up of proteins and RNA. Okay, it is made up of proteins and RNA. We can show it in diagram. See, I have indicated this DNA, right? DNA, these indications, they are genes. So we indicated these are the portions of the DNA and they are made up of proteins or RNA. Okay. Now few important features in case of transcription. It is a highly selective. Just now we discussed that in case of transcription, only some or groups of genes are being transcribed. So complete DNA is not transcribed. Okay, dear students? Clear? So it is very selective process. Only some parts will be transcribed. And the sequences some sequences will mark the beginning and some sequences will end the process. And these signals, they are controlled by the enzymes. So when to start and when to stop, this process is highly controlled. Second, many of the RNAs transcripts are synthesized as precursors as primary transcripts. Means once you've got the mRNA made, then there are certain modifications which will be done in the mRNA. So convert it into functional mRNA. So it is not a necessity that RNA that has been made is 100% functional. It may be functional, it may not be functional. So it is a basically primary transcript which is made. Third is, so as I said, few modifications, trimmings may be required. Now transcription, it occurs at fixed positions in the cell. Okay, it occurs in fixed positions in the cell. Like in case of prokaryotes, it only occurs in the cell cytoplasm of the cell. So in prokaryotes, it will occur in the cytoplasm of the cell. All the RNAs, they will be made in the cytoplasm. In case of eukaryotes, it will be done in the nucleus and mitochondria. Okay, it will be done in case of nucleus and mitochondria. While rRNA will be made in the nucleolus, tRNA and mRNA, they are made in the nucleoplasm. So we have given the further division also, but you must remember, there is no need to remember this one, but you need to remember this, that in case of eukaryotes, the transcription process occurs in the nucleus and the mitochondria. Now see, I have a statement, and this statement was 2 years back, it was asked in the board exams. The statement is, in transcription, only a segment of DNA or only one of the strand is being copied. Why? What is the reason for this? So first you understand the question. I'm again repeating. ट्रांसक्रिप्शन के केस में सिर्फ डीएनए के कुछ सेगमेंट्स ही ट्रांसक्राइब होते हैं और डीएनए का सिर्फ एक ही स्ट्रैंड है जिसे कॉपी किया जाता है. ओके, डीएनए इज मेड अप ऑफ डबल, it has a double helical structure, so it has two strands. So why only one strand is copied? So dear students, I'll be discussing the answer. The very first point of this is, if both strands are copied, now suppose we are DNA is separating and both strands are being copied, so we will get two RNAs. Okay, because both will act as a template, so we will get two RNAs, and these two RNAs will have different sequences. Both of them will have the different sequence, so they will produce different proteins altogether. तो हमारे पास दो स्ट्रैंड मौजूद होंगे आरएनए के और दोनों आरएनए से हमारे पास अलग अलग प्रोटीन्स बनेंगे. And that will create a problem because a lot of proteins are being synthesized in the cell itself. Although that much of the proteins is not required. So this could be the one reason. Second is, now suppose this is your DNA. When both will separate and they will produce, you can say, let's take another color. If both will separate, then two RNAs will be produced. Like one will have this and second will also have this kind of. And these two RNAs will have complementary sequences. Okay, they will have complementary sequences. Why they will have complementary sequences? Because the parental strand also has complementary sequences, isn't it? So there are chances that these two RNAs, when they are removed, they may get entangled with each other. So they may join with each other and form the DNA rather than forming the RNA. तो possibility है कि दो RNAs जिनकी coding जो है complementary है, dear students, they may join with each other and they will form the DNA ultimately. And what we require here? We require the RNA so that the proteins can be made. Okay. Now your transcription unit. Now these are the basics that I am talking about. Only then we can talk about the transcription process. So your transcription process or transcription unit contains three regions. One is a promoter region, another is a structural gene, and third is a terminator region. Promoter means from where the process will start. Then the structural genes, those will be helping in the process. Terminator means where actually this process will be stopped or terminated or finish. Okay, so here the process will start, here the process will finish, and this is the process, meantime the going on process. The structural genes, right? Now I have shown you the location. This is a schematic structure of a transcription unit. Just look at it. First of all, we have two strands, isn't it? One I have shown with blue, another is with green. This is a diagram that I have taken from your NCERT itself. In fact, the whole content is from NCERT. The only bit is that it is placed separately. So I'll be telling you where it is. So DNA has two strands. The blue is running from 3 to 5 prime and the green is running from 5 prime to 3 prime. So the one which is running from 3 prime to 5 prime will act as a template strand. Means this strand will be coded. Okay. This strand will act as a basis so that RNA can be produced. So that is why we call it as a template strand. While the second strand indicated with green is called a coding strand. Now this strand is of no use in transcription. But still it is being coded strand. Why? Because the mRNA which is produced in the center, जो mRNA produce होगा, right? Suppose this mRNA is being produced. This mRNA will have the sequences exactly same to this sequence. ठीक है? जो mRNA हमारा produce होगा उसकी sequence बिल्कुल exactly 5 prime 3 prime coding strand के साथ match होगी. That is why we call it as coding strand. Okay? Clear? So we have a promoter. You can see the location. Promoter is present. Now any reference that we'll talk about will always अगर हमने लोकेशन बतानी है किसी चीज़ की तो हम कोडिंग स्ट्रैंड के रेफरेंस में बताएंगे. ओके, सो प्रमोटर इज प्रेजेंट एट नियर टू द 5 प्राइम रीजन and terminator is present at 3 prime region. In between you have your structural genes which will help in this process. Okay, so I hope you've understood the transcription unit. The transcription unit has 3 things: promoter, structural gene and the terminator. Template strand means the strand which will act as a template. That will be called as template strand. The coding strand, it is of no use in transcription, but the mRNA which is produced will have exactly same sequences like the coding strand. Now let's see. So we have one template strand, another we call as coding strand. Let's see how they function. Okay, let's see how they function. Now देखिए, this I have indicated a portion of DNA. Okay, this I have indicated a portion of DNA. So this 3 prime to 5 prime will act as a coding, the template strand, right? 3 prime हमने याद रखना है कि जो strand 3 prime से 5 prime है वो हमारा template strand होगा. Okay, so this will act as our template strand. अब देखिए sequences they are given: cytosine, adenine, guanine, thymine. Okay, C is cytosine, A is adenine. I hope you remember the bases. T is thymine, G is guanine. Okay, so against C we will have G, against A we will have U here. Now in case of mRNA, instead of T we will have uracil. Okay, I'll write here, we will have uracil. Now in case of mRNA, we will not have thymine, we will have uracil. Okay, so we will have uracil. Clear? So C, against C we have G, against A we have U, G, C, T we will have A, again U. So in this manner, the opposite bases will be added. Now if you look at this sequence, इस sequence को आप 5 prime से compare करिए. It is exactly same, but there is only one difference, that instead of thymine we have uracil. Okay, so in case of RNA we do not have thymine, we have only uracil. Now the functions of RNA polymerase. This is RNA polymerase. Now your RNA polymerase, it performs a lot of functions. First of all, it searches and binds the promoter. Then it opens the double helical structure. Then it selects the ribonucleotide which is to be added. Okay, it will select it will check which ribonucleotide or which base has to be added. Then again, it also terminates the process. It will terminate the process, isn't it? It will also interact with the activator and the repressor. तो RNA polymerase बहुत important enzyme है in case of transcription. This will only identify the promoter. This will only identify the activator and repressor proteins, and this will help in adding the sequences or the bases in the elongation process. Okay. Now, most important, your RNA polymerase, it only works in 5 prime to 3 prime direction. ठीक है, अगर आपको याद हो तो DNA also works in 5 prime to 3 prime direction. So RNA polymerase can only work from 5 prime to 3 prime direction, and that is why the strand which has a polarity from 3 prime to 5 prime will act as a template strand. Only this strand can act as a template strand, right, dear students? So the strand which is moving from 3 prime to 5 prime will act as template strand, as RNA polymerase can only act in 5 prime to 3 prime direction. Now this will be the homework for today. First of all, you will make notes of the topic and you will read the page numbers 107 and 108. This information that I have talked about is given on this page 107 to 108, introduction to transcription. Okay, dear students? So I hope you will complete, and if there is any doubt, you will send your doubts or get your doubts cleared by me. You will talk to me, you will correspond with me. Okay, dear students? Okay, bye everyone.